Hello students, welcome to EPG Pathshala. I am Dr. Pooja Gulati from Department of Microbiology, Maharishi Dayanand University, Rohtak. Today we are going to talk on the module Nucleic Acid Based Assays under the paper Animal Cell Biotechnology. So students, let's see what we are going to learn from this module. Firstly, we'll know about the importance of nucleic acid-based diagnostic methods. Secondly, you will know about the molecular diagnostic techniques. Then, most important is that you'll come to know about the principle and the methodology of the various molecular methods and compare the methods based upon the advantages and disadvantages. As all of you know, in the last few decades, the field of molecular diagnostics has advanced tremendously and rely on novel molecular techniques for early and easy detection of microbes, especially pathogens. These nucleic acid-based assays have helped in the easier, fast and timely detection of viral as well as bacterial load in clinical samples. They have enabled detection and identification of fastidious, contagious, and even unculturable microorganisms, which are very difficult to diagnose without these methods. Molecular techniques have emerged out as highly sensitive, specific, accurate tools for the clinicians, and these methods they use probes which can be broadly characterized into non-amplified nucleic acid based probes and amplified nucleic acid based probes. Nucleic acid hybridization assay it forms the basis of these techniques, and in this technique I'll just brief you about the nucleic acid based, how does the nucleic acid based hybridization is carried out. The nucleic acid hybridization utilizes alleling of complementary sequences to form a stable structure, which is actually a hybrid for detection. The unique ability of a DNA to bind only to its complementary sequence is utilized in the hybridization technique. In this technique, the probes are labeled single-stranded sequences which are complementary to the target sequences, and thus they bind to the target sequences by complementary base pairing. In hybridization, we can use either DNA or RNA sequences whose presence is to be detected in a cell or an organism, and these sequences are referred to as target. For stable hybridization, following points should be observed. First and the foremost is probe and its size. An ideal probe is a single-stranded nucleic acid which hybridizes to the target sequence. The probe can be DNA or RNA. The size of the probe can range from 10 to 10,000 nucleotide bases. Usually, a probe consists of 20 bases. Since short probes are easy to prepare, more stable, hybridize rapidly, and are able to detect minor changes in the target, they are most commonly used as suitable probes in different assays. The second point to be taken in mind is specificity of the probe. The basic sequence of the probe defines its specificity. The probe should not self-hybridize or bind to a non-target sequence, but should be targeted to hybridize to specific target sequences only. The target sequence should be chosen which is not present in non-target cells. Labeling of the probes. These probes should be easily detected after hybridization. The label should either be attached or incorporated within the probe. The isotope of phosphorus, sulfur, and iodine are commonly incorporated into the probes, while the enzymes like alkaline phosphatases, horseradish peroxidases are covalently linked to the probes. Another probe involving biotin is commonly used where enzyme-labeled evidin molecules are used for biotin detection. The fourth point is choice of the target nucleic acid, which is very important in the identification of the pathogens. Various targets have to be chosen for probe development, including detection of genera, species, and in some cases strains as well. Generally, conserve sequences targeting a common or same nucleotide sequence found in several genera, like ribosomal RNA, which is present in different families of microorganisms, is used as the target sequence. Non-amplified nucleic acid hybridization assays. The non-amplified hybridization assay can be achieved in three ways: liquid phase, solid phase, and in situ hybridization. First, we'll take liquid phase hybridization. The target nucleic acid and the labeled probe are allowed to interact in an aqueous environment, hence it is called as liquid phase hybridization. The hybridized double-stranded DNA can be detected using trichloroacetic acid precipitation, using acridine ester labeled probe which hybridizes to the target sequences. On addition of H2O2, the duplex will emit light which is measured by luminometer, or using hydroxypeptide columns which selectively bind to double-stranded DNA. This technique has been employed in detection of various bacteria like Mycobacterium, Streptococcus, Staphylococcus, and many other. Now let's discuss about solid phase hybridization. Solid phase hybridization is more easy method to perform as compared to the liquid phase hybridization. In this method, the target nucleic acid is bound to a surface and thus it is called as solid phase hybridization. The solid phase used here is nitrocellulose or nylon membrane. So our target molecule is linked onto a solid matrix. On hybridization, with a probe which is in a solution form, the hybridized target nucleic acid can be easily measured by color development, fluorescence, luminescence, or radioactivity, depending upon the label which is attached to the probe. This method is most commonly used method in the clinical laboratory, and the solid phase hybridization in the form of dot plot and colony plot can be easily used in the clinical laboratories. However, this technique is time consuming, which limits its practical application in the clinical laboratory. The following steps describe the basic principles of dot or colony hybridization. The sample containing cells is captured or blotted on a solid support such as nitrocellulose filter disc. These cells are then disrupted to obtain double-stranded DNA bound to the solid support. The double-stranded DNA is denatured to obtain single-stranded DNA which will bind to the solid support. The filter is then washed and unreacted sites are blocked. Labeled probes are added and allowed to hybridize to the target sample. The unbound probe is washed off and the hybridized probes are detected. Although this method is the oldest, but it is prone to non-specific background interference. In situ hybridization. This in situ hybridization involves the hybridization in the cell itself. So the target nucleic acid is found within the cell or the tissue fixed onto the microscopic slides. Thus, this method will indicate the intracellular localization of a gene expression in a cell. It is commonly used for tracing viral sequences, proteins involved in replication, etc. The most common of in situ hybridization is fluorescent in situ hybridization, which uses fluorescently labeled probes and fluorescent microscopy to detect bacterial species within the clinical samples. Amplified nucleic acid based assays. In this technique, the detection of a bacteria or the viral target is achieved by amplification of the target, probe, or signal molecule. First and the most commonly used method is the target amplification method, as you must be all aware that PCR was the first method which was used for the target amplification by Gary Mullis. In the target amplification technique, basically, an enzyme-mediated assay is used where the synthesis of the target nucleic acid is achieved with the help of the specialized enzyme. For example, in the case of PCR, we use Taq polymerase. So first, we will discuss the most important technique of PCR, as this technique has allowed the rapid and specific identification of many pathogens in the clinical laboratory. PCR amplifies and produces millions of copies of target DNA sequence within a few hours. In general, a PCR reaction mixture will contain 2 oligonucleotide primers, a target sequence, which is the DNA from which you have to identify that particular sequence, then DNTPs, that is deoxyribonucleotide triphosphate, which will be used as a substrate by the enzyme Taq DNA polymerase. Then in addition to these main constituents, we also add MgCl2, KCl to these reactions to increase or decrease its specificity. And all this reaction is carried out in the 1X reaction buffer, which is provided with the enzyme which is used in the reaction mixture. The primers for identification of the pathogen are generally 18 to 25 base pair long oligonucleotides, complementary to the sequences flanking the target sequence. So the primers are designed from both the sides, one from the forward and other from the reverse, which are through the flanking regions of the complement of the target sequence. Now I will briefly discuss the steps which are involved in the PCR reaction. The first step is denaturation. Double-stranded DNA melts, resulting in single-stranded DNA, which can act as a template for primer binding. The denaturation is generally carried out at a temperature of 95 degrees Celsius in a thermal cycler. Annealing, the primers are in this step, the primers, they bind to the single-stranded DNA. The annealing temperature varies depending upon the primer's GC content. Generally, after denaturation, annealing is performed at 40 degrees Celsius to 68 degrees Celsius depending upon the annealing temperature of the primers. Following this, extension is carried out. During extension, the Taq polymerase enzyme uses DNTPs to synthesize new DNA fragments complementary to the template. For this, the temperature of 72, which is an optimum temperature of Taq polymerase, is used. If other polymerases are used, then their optimal temperature is used for the extension. These cycles of denaturation, annealing and extensions are repeated at least 25 times to 35 times, varying upon the size of the product, and thus in the end the amplicon is detected by electrophoresis on the agarose gel. This method has various applications. It is used for structural analysis of genes like Shiga toxin producing genes in E. coli and mutational studies in genes like disease-causing mutations in COL1A1 and COL1A2 genes of type 1 procollagen. Then PCR is an effective tool in genetic fingerprinting. It represents a fundamental technology in major sequencing products like Human Genome Project. Anti-bioterrorism tool, it can act as an anti-bioterrorism tool by detecting a number of category A bioterrorism agents, which include variola major, Bacillus anthracis, Yersinia pestis, and Francisella tularensis. Then can be used for diagnosis of various inherited diseases like cystic fibrosis, muscular dystrophy, hemophilia A and B, and many others. Several modifications of PCR are routinely used in the clinical laboratories, and amongst this, the most important is RTPCR or reverse transcriptase PCR. This method allows amplification of RNA molecules and utilizes reverse transcription to produce single-stranded cDNA from RNA, which is further used as a substrate for amplification. Thus, this method can be easily used for detection of RNA viruses, and the major advantage of this method is that it is a sensitive approach than Northern blotting for RNA-based detection. Another variation of PCR is nested PCR. It is a more sensitive technique. It uses two pairs of primers for the amplification of target sequence with 2 rounds of amplification. The amplified product of the first outer primer is used as a target for the second round of amplification with the internal primer pair, thus making this technique more sensitive than other PCR method. However, the major disadvantage of this is high risk of contamination, and it is a time-consuming assay. Multiplex PCR is also an important tool for the clinician as it targets multiple sequences with 2 or more primer pairs for simultaneous detection within the same reaction mixture. The primer pairs must have similar annealing temperature and lack complementarity. It is used for detection of viral and bacterial pathogens like herpes viruses, respiratory viruses, where spirogroup specific primers are used in Neisseria. Multiple foodborne pathogens can also be detected in one go using multiplex PCR. The most important advantage of this method is that it is rapid, sensitive, and cost-effective approach for detection. Now let's discuss real-time PCR. Real-time PCR is a very important technique as this variation of PCR allows quantification of the sample. So it is a quantitative PCR and can measure the amount of amplicon produced as the PCR reaction continues. Thus, both amplification and detection of the target sequence occur simultaneously. The amplification plots which are produced in the PCR will show the increase in the fluorescence with every cycle. Various specific and non-specific probes are employed in this method. For the non-specific probe, the most commonly used probe is cyber green dye, and the specific probe is fluorescent label probe, and these two techniques I have discussed briefly in the modules. Real-time PCR using cyber green dye. This is the easiest and less expensive method. Cyber green specifically binds to double-stranded DNA, intercalating between bases and fluoresces only when bound to DNA. Fluorescence signal is detected after every cycle, either at annealing or extension step. Thus, as the PCR proceeds, amount of the signal obtained will keep on increasing and will give an estimate about the amount of the RNA or DNA which is present in the sample. But the major disadvantage of this method is that cyber green detects any double-stranded DNA non-specifically. Thus, reaction mixtures should be such that only a single gene-specific amplicon are produced without any non-specific secondary products. Real-time PCR using label probe. This method is more sensitive method as compared to the cyber green method. Here fluorophore label probes are used which bind to double-stranded DNA molecule. The probe is an oligonucleotide having a reporter fluorophore dye and an acceptor quencher dye attached to its 5' and 3' end respectively. In the presence of the target sequence, the probe binds downstream to one of the primer site, and on extension by Taq polymerase, the probe is cleaved by its 5' nuclease activity. The fluorophore thus gets separated from the quencher and emits fluorescence. So amplification plots can be produced which will show increase in fluorescence with every cycle. Some of these labeled targeted probes are Tachman probe, which is shown in the figure, molecular beacons and scorpions, and Fred probes, which are commonly employed in the laboratories for real-time PCR. The target-based amplification techniques, and now we will discuss the probe-based amplification technique. In probe amplification method, only those target sequences are amplified which contain the probe sequence. And for this, the most important method is ligase chain PCR. Two pairs of synthetic oligonucleotide probes are used, annealing close together on the complementary target sequence. The gap between these oligonucleotides is filled with thermostable DNA polymerase, while the nicks are sealed with the help of thermostable ligase. The ligated products serve as a template for the next amplification cycle. This technique is used in the detection of viruses like human papillomavirus, herpes simplex virus, and HIV, and many bacterial pathogens like chlamydia, trachomatis, Neisseria, gonorrhea, etc. The major advantage is that it is a two-step method, and thus it is more specific than the PCR used alone. Cycling probe technology. The cycling probe technology detects the presence of target nucleic acid sequences without target amplification. Here, the reaction is carried out in isothermal conditions where a chimeric probe, that is DNA, RNA, DNA, binds to the target nucleic acid sequence. On hybridization, RNase H cleaves the RNA internucleotide linkages, dissociating the probe from the target and making it available for the next cycle. The cleaved products can be measured by various techniques like electrophoresis or using fluorophore labeled probes. The cycling probe technology has several applications, like it is used for the specific assay for antibiotic resistant organism and DNA and RNA viruses. The major limitation of this method is that it is time consuming assay and lower amount of the target sequence, and the higher amount of RNase H is required for the assay. Slide 23, signal amplification techniques. This assay identifies the desired nucleic acid by amplifying the detectable signal sufficiently. Several assays utilizing signal amplification for detection of target nucleic acid are non-isotopic test detection system, which can use acridine ester labeled probes, phenyl phosphate substituted dioxetane or enzyme labeled antibodies, or it can be nucleic acid based method for the signal detection. In addition to these, several newer diagnostic methods have come up which have really helped the clinician in the accurate and fast detection of the pathogen. The most important of this technique is microarray. In the microarray technique, DNA fragments are synthesized and immobilized as spots on the silicon slide or chip in a specific order. These microchips are on probing with the target molecule result in either quantitative gene expression, means either quantitative or qualitative data. The quantitative data means you can use the various dilutions of the sample and you can check the amount of the gene expression, and then qualitative means you just detect the which pathogens are present or not. Different types of microarrays have been developed, including printed microarrays, in situ synthesized oligonucleotide microarrays, high density bead arrays, electronic microarray, and suspension bead arrays. The microarrays have several applications. They are used for detection of bacteria and their identification, microbial typing, and gene expression profiling. The major disadvantage of this method is that you can study several different gene expressions simultaneously. But the disadvantage of this method is that heterogeneous clinical samples are difficult to analyze. Differentially expressed genes might not be detected efficiently. In addition to the techniques which I have just discussed in this module, there are other molecular techniques also which are used for the identification as well as typing of various pathogens. These techniques are based upon sequencing and enzymatic digestion of nucleic acid. Sequencing is performed to determine the order of the nucleotides which are present in an sequence, and this can be done only in specified laboratories, so it cannot be used in every laboratory, and it requires a lot of cost, so it is a cost intensive or expensive process. In addition to this method, the method which can be easily employed in the laboratory is based upon enzymatic digestion of the nucleic acid. And for this, the most commonly used method is restriction fragment length polymorphism, or commonly known as RFLP. RFLP, they determine the variation or polymorphism in a nucleotide sequence to differentiate between two sequences. This technique uses restriction enzymes to digest the DNA at a particular site, and then the digested products are run on the gel. However, the bands which have small differences or minor differences in the sequences in the base pairs, they cannot be differentiated by this method. So another variation which is used is pulse field gel electrophoresis. Pulse field gel electrophoresis is a gold standard technique which is used for molecular typing. Here the electric fields are applied from different angles, separated large DNA fragments. Hence, resolution of the fragments differing in the few bases can also be achieved. Then another method which is commonly employed these days for molecular typing is ribotyping. In ribotyping, restriction enzymes are used to digest specific rRNA coding genes. For example, if it is bacteria, then 16S rRNA gene is used. If it is fungi, then you have 28S rRNA which can be used for the sequence, for the ribotyping. And after the digestion is carried out, a gel is electrophoresed and the sample are electrophoresed on the gel, and the result can be seen. This method helps in subtyping the bacteria beyond the species and the subspecies level. So students, let's now summarize what we have learned in this module. In this module, I have told you about the principle, protocol, advantages, disadvantages of various nucleic acid based methods. Nucleic acid based techniques that have been commonly adopted or in the laboratory over cultural techniques have been discussed. Various amplified and non-amplified nucleic acid based methods which are employed for the early detection of viruses and bacterial species are also discussed in this module. Then hybridization assays such as liquid phase, solid phase and in situ hybridization has enabled detection of viruses like human papillomavirus, cytomegalovirus. With the advent of PCR and its various modifications, molecular diagnostics have brought new dimensions to the scientific world. And on automation, these methods will be both cost effective and time effective. Thank you.